Let's get started. All right, so thanks everyone for being here, and welcome to the Center for Reproducible Biomedical Modeling seminar series. We are happy to be starting this back up after a slight pause, with this as the inaugural first seminar of this next series. We're going to be focusing on a few topics throughout the year. Starting off with multi-cell modeling. We'll be touching on topics such as annotation and credibility as well. And so today we bring to you two experts on multi-cell modeling, TJ Sego from the University of Florida, James Glazier from Indiana University. These two have worked on new methods and software for multi-cell modeling, which makes these models more accessible and reproducible. And well, we'll hear what they have to say about that. So today we're going to start off with TJ. I asked them to prepare a 20-25 minute talk. I'll be moderator to make sure we're all on time. So we're doing TJ first, then James. Please save your questions for the end, and we'll have some time at the end for discussion. So without further ado, TJ.
Great. Thank you very much for that kind introduction, and very happy to be talking with all of you today. I'm going to assume that you can see my screen and hear me okay. Someone speak up if that's not the case. Otherwise, yeah, very, very glad to be talking with all of you today. So James and I have been coordinating our talks today to try to give you a broader scope of what's going on in the world of multicellular and multiscale modeling. So we're going to start in my time with looking a little bit closer at some of the specifics of what some of these models tend to look like, and just a very, very brief glance at some of the guts of some of what current multiscale multicellular modeling looks like. And then we'll hand it off to James, and James will take us into a much well, James can explain himself, so I'll just carry on. Yeah, so I want to start this just by kind of centering us on, at least in part, like where a lot of my interests tend to be in the world of multiscale modeling, and that is to take what I tend to think of as sort of like classic systems biological modeling, something like a network of the cell cycle, like what you can see here in the bottom center of my screen. Take network modeling and put that modeling into a multicellular heterogeneous tissue context. And to motivate this a little bit, you can find lots of models of the cell cycle. This is a very well-studied system, lots of active research in the world of the cell cycle, but most modeling is done within the context of considering a single cell or a homogeneous population of cells, each of which is assumed to behave approximately the same. And there are lots of useful applications for that approach. But then when we look in all sorts of relevant developmental or medical contexts, we find that, of course, like just about everything else in biology, it's much more complicated than that. And there are a couple of visuals here shown here on my screen to help you give a sense of that. So this is here in the top right. So this is imaging from the second mitotic wave in Drosophila. And in particular, we're looking at this sort of classic spatial patterning of delta notch signaling. And for those of you who aren't familiar, we'll look at this a little bit more here in a couple of minutes. But in general, this is a highly conserved signaling pathway, and generates spatial patterns like you see here, where you find alternating cells that are expressing different types of signals. And this really only can be modeled with spatial information. And now what's interesting here in the terms of something like cell cycling is that there are lots of aspects about delta notch signaling that's highly spatially heterogeneous that affect the cell cycle. And here in the bottom right, so this is a figure mapping out recovery in the pulmonary alveolus. And this is another set of events where cell cycle obviously is quite important with proliferation for wound healing. And there are a lot of aspects about this that have lots of spatial information and heterogeneity involved. One being delta notch signaling with intercellular regulation by cells within the alveolus, but then also signaling with release of growth factors from the extracellular space and the environment. There's release of growth factors that also can induce and promote cell cycle in cells. Now, the distribution of these chemical species is also potentially highly spatially heterogeneous. This is all to say that while these networks are important and they certainly have their place, there's also a place for considering how these types of networks impact the spatial organization of tissue, its maintenance, its repair. And this is a lot of where multiscale multicellular modeling is going, is currently working, has been working for a while now. But this tends to be sort of standard practice in a lot of applications. For me, with viral infection modeling and immune response, lots of different applications where taking a network model and putting it in a multicellular context can explain lots of interesting things about both the networks and their role in heterogeneous tissue. So here's an example of this shown in the bottom right. So this is an animation from the multicellular, multiscale simulation package that James and I worked on, CompuCell 3D. This is a simulation of combined delta notch signaling and cell cycle for wound healing. And what you see here is something that looks like a typical simulation using the cellular POTS model, which is the cellular dynamics method underlying CompuCell 3D. You have spatially explicit cells. Each of these is modeled individually using agent-based modeling principles and the statistical physics of the cellular POTS model. But then there's all the colors that you see. This is the same cellular distribution plotted in all of these different subplots, but each of these is showing different states of this network model that is attached to each individual cell. So the same type of network modeling, in this case, this is representing the cell cycle using stochastic Boolean network dynamics with MABOS. The same type of modeling, except those dynamics are being coupled with the agent-based cellular models according to CompuCell 3D. And so what we tend to do then is to take the multicellular agent-based models that have been developed and worked on for quite a few decades now, like with CompuCell or with other simulation platforms, but to start to incorporate the network dynamics that we attach to individual cells and use those cell state dynamics models to drive the agent-based models at the multicellular scale. And likewise, to provide feedback from the multicellular models back to the subcellular network models. So to try to conceptualize this a little bit, now this could be with ODE models, Boolean network models. The underlying method for cell state dynamics that we tend to work with is mostly unimportant here. What's important is something like this sort of rough mock-up here that I've shown to the right. So imagine that this block here is something like the state space in which you could find the state of a cell in a particular model at any given time. Each of these triangles just shows some particular state in the state space of each of the cells in a simulation. And for Boolean network models, this could be some Boolean state. For ODE models, this could be something like just a state vector of all of the variables in an ODE model. But what's interesting here in terms of embedding these models into multicellular modeling is to start to think of those states as working their way through decision-making processes about the cellular properties and processes of what each cell does. So for example, you have something like a Boolean model with some particular state, you can assign that to being either something like an epithelial cell type, which is something that's very familiar to multicellular agent-based models. We work with all sorts of cell types for each of our agents. You can think of a state as falling into the domain of being either an epithelial cell type or a mesenchymal cell type. And based on where we find our state, according to the subcellular embedded models in each individual cell, we can then assign corresponding properties and processes in our agent-based models. And this can get quite detailed. So depending on the particular application and what models or what processes you may be interested in modeling, you can start to write network dynamics and attach those to cells and connect them with the environment to describe increases in volume due to intracellular state, have things like mitosis, and of course, more interestingly, at least I think, for doing this in a multicellular context, you can also start to provide feedback for those subcellular models from the multicellular models. So if we jump back real quick to our animation with this CompuCell simulation, we look at each of these individual cells at every simulation step. We can ask things like, what cells is our cell in contact with? If we had diffusive signals here for like reaction diffusion dynamics, we could also ask what concentrations of different species is our cell being exposed to. Now, if we jump back to where we were, we can then take that information at each simulation step and use that as something like updated boundary conditions or inputs to our embedded intracellular models. Now, to give you a sense of what this tends to look like, at least for CompuCell, so this code snippet here in the bottom right, this is a code snippet from the actual implementation from that simulation that we've been looking at. And custom model specification in CompuCell is done in Python. And so you see some Python code here. And we can just walk through this real quick. So this particular function is called at every simulation step. And all this model does is it just goes through every single cell in a simulation, gets the Boolean network that's attached to that cell, called them MABOS models, because that's what they are in this case. And for this particular simulation, to implement the interaction of networks through contact mediated interactions for delta notch signaling, this then just goes through and asks CompuCell, what cells is my cell attached to? And then tell me what expression of delta is in each of those cells. And I'll use that information from the neighborhood of each cell that I'm looking at to update the information in the Boolean network for the cell that I'm considering. Now, again, this can be arbitrarily complex. The same sort of information could be retrieved in terms of chemical species. This could take information from a subcellular model and implement changes to cellular properties, or say, induce like release of different species or differentiation, so on and so forth. It can get pretty complicated pretty quickly, but this is the basic premise of the sort of cell-centric multi-scale agent-based modeling that a lot of us in this community have been doing in the recent past. Now, all of this is certainly not without its challenges. James will go into more detail about this, and so I've reserved this space to just communicate a few things here that are of particular interest to me and that I think are especially important. So a lot of the implementation of these technologies can get quite complicated, if not impossible. So in this example that we've looked at, this capability of doing stochastic Boolean networks in CompuCell, this is maybe a year old. This is a newer capability for that platform. There's longer running support for integrating libRoadrunner into CompuCell, so doing ODE models attached to individual cells or describing dynamics outside of a spatial simulation. That's been around for a few years now, but what's important here is that it is a lot of work potentially to integrate one simulator into another, if it's possible at all. A lot of simulators that are commonly used are simply just not designed for integrated applications like this, which is essentially what this is. For doing ODE models and stochastic Boolean network models within CompuCell, this is an integrated application for each of these simulators. And a lot of simulators are just not designed for that. CompuCell was only had that support beginning last year. And so if it's even possible to do this with different simulators, it can be a non-trivial amount of work. And even after presenting that capability to the community for particular research applications, it is then just sort of like the beginning of the well of problems. So even just translating data from one simulator to another is often not well defined, if at all, because each of these simulators uses different methods, and often it's not clear at all how to translate data from one method to another. In a stochastic Boolean network, we have either high or low states for different nodes in a network, and what exactly do those values mean in a multiscale multicellular model? Same with the ODE models. What does an actual value in an ODE model mean in terms of a multicellular agent-based model? Breaking up models into modules is also quite difficult, if not just a very subtle art that eludes most. And much of that is to do with that most of the modularity that we want to believe exists in biology is model specific. It's very rarely clear how exactly it's best to break up biological systems into modules that could be interconnected. And most often, at least in my experience and by my observations, this is almost entirely context specific. And pair with that for most methods that are commonly used, whether it's at the multiscale or multicellular scale, with the cellular pots model or with center models, all sorts of methods out there, but very few of them have standards, like what ODE modelers enjoy with SBML. For most methods, there is no standard platform agnostic way to describe these models, which makes model sharing extremely difficult. It throws reproducibility out the window in almost all cases, and really makes leveraging any sort of modularity that we at least imagine, makes it very difficult to use, except in the platforms in which those model modules are defined. And then lastly, and I'll turn it over to questions. So coupling these kinds of models across different scales introduces a lot of difficulties in terms of the numerics, with computational performance versus accuracy. So with these embedded models, for example, for describing what's going on inside each individual cell, the rate at which those processes occur is generally much faster than the time scale at which we tend to do multicellular dynamics. So in an ODE model, we can do integration on the order of milliseconds, seconds, something like that. Whereas with the multicellular models, we tend to operate more on seconds to minutes to hours. And those differences can be really important for different kinds of models. For example, if I have a subcellular or intracellular model that has dynamical features that are very fast, I have the choice of either integrating at the sort of the scale of my intracellular models, in which case my multicellular scale models will take an extremely long amount of time. Or I can integrate more at the scale of the multicellular model and potentially miss the dynamical features that occur in the intracellular systems between simulation steps. So it's not clear how exactly to deal with these issues in a standard way. I think a few others have some ideas about how to do that, but At the end of the day, there's really no good way to really address these issues when combining these models, except sort of ad hoc, solutions that end up in the supplemental materials of papers that tend to get forgotten. So there's lots of interesting challenges here. With that, I'll turn it over to questions or yeah, however we want to proceed. Thank you very much.
Let's go to James and do all the questions at the end.
Sounds good.
James, you're muted. Maybe we could have gone to questions.
Oh, sorry about that. Screen share, okay? Is the screen share all right?
Yeah,
It
we
is,
can see
yeah.
you.
Okay, thank you. Sorry about that. My computer is having a little glitch with the screen, the built-in screen. So, okay, I want to thank you for the opportunity to talk. This is a big topic. I'm only going to talk about a few subtopics. Many of you will be familiar with aspects of slides, recognized slides that have been shown for years by now. And also, people who were at the Combine meeting will see that this is essentially a cut-down version of the Combine talk. Trying to understand challenge and opportunities for multicellular models is really pretty hard because the range of problems that we're working on is very large, and we have to think about a rather complex set of physical and biochemical interactions, and the kinds of questions we're asking are very diverse. Typically, we break our problems up into these three levels of some kind of intracellular network model, some description of cells and their behaviors, and some macro description of the environment of the cells, and possibly another non-cellular description of the transport in the environment around that. You might have additional layers, of course, beyond that, either inside cells or outside. But the main problem that we have to address, which is a little bit different perhaps from what you face when you're doing network models, is that our interests are also an emergent agent or emergent phenomena from agents, are also mechanism driven, but our outputs are fundamentally movies. And things are based primarily on quantitative submodels of object behaviors and processes. And a lot of these are existential processes, which you don't typically get in network models. You create cells, you destroy cells, cells change their interactions, as you saw in the movies that TJ was showing. And of course, we come back to the question of why are these kinds of models not more influential after 30 years of working on them? And I think that the lack of shareability and reusability is a big part of that. I've had this slide since 2009. This is a famous experiment that was done by Keller, where they're able to track every nucleus in a developing zebrafish embryo. That should be tremendous data for building models of embryology, but we are not yet able to take that kind of experimental data set, build a digital twin of it, and run that in register. And if we're ever going to be able to do that, we're going to need to be able to do standards-based and component-based model specification. These days, cell atlases are very fashionable. Chad Zuckerberg is just, of course, is one of the many organizations that's put a lot of effort into that. Those are dead samples. One would like to be able to take a cell atlas and be able to populate it with cell types based on the omics information for each cell, and then build dynamic models based on that. What would it take to be able to do that? And finally, you'd like to be able to integrate these kinds of models into a workflow that generates knowledge. And you don't want to strand the information in these complex models in code. And so you then want to be able to build components of some kind that are reusable and implement the knowledge that is intrinsic to these kinds of problems. These are all difficult problems. We're not certainly going to talk about all of them today, but I think it's worth thinking about the whole workflow of knowledge generation and application because it's going to inform the kinds of decisions that you make at each scale. We want to be able to build virtual tissue models, multi-cell models, quickly that are grounded in experiment. That means we need standards, and it's not easy to do. TJ's talked about the issue of software interface and capability standards. Perhaps today it's better for me to focus in the limited time we have on model specification standards. In particular, I think the key, and this is something that is still not a solved problem even for network models, is the specification of interoperable model components that allow you to reuse elements of model again and again. At the moment, if we have two network models specified in SBML, and they both, for example, involve ATP, there's certainly no guarantee that if we pull those two network models, run them together, that they'll work. And that ability to compose models, if you want to think of it mathematically, or interconnect model components is something that is taken for granted in a lot of disciplines, but not at the moment in ours. And so I think it's worth coming back and asking the question, what would it take to build a library of reusable instantiable biological components that we could use to populate an experimental image and turn it into an executable model? Well, it's not easy. And if we look at the situation, we understand why. These are multi-scale models. There are a lot of issues that we have to deal with that are complicated. For one thing, there's a lot of heterogeneity at different scales, and the degree of resolution that we're going to use to represent those scales will change what parameters and information are exposed. And that means that there isn't an agreed upon API at each scale for saying how our components are going to work. We have to be able to connect these across scales. We have to be able to deal with different solver technologies within and between scales. We have a lot of programming environments. Of course, we've used CompuCell for years. And the more I work with CompuCell, I think the more I feel that maybe CompuCell is solving the wrong problem. That maybe the idea of making it really easy to write Python code is not the way we need to address building models at this point. One of the complexities that we face is that we don't know a priori how these models are going to be used. These are all CompuCell simulations. Two of them are running, well, these are movies in 3D, but you see that there are a whole variety of different morphologies, questions, processes that are mapped. We have to be able to be flexible enough to be able to encompass all of these different questions in a single formalism. These are yet more examples of CompuCell simulations. Some of them come from the EPA and other groups. And so in this case, on the top left, we have a lot of subcellular detail in the simulation. Other top center left is a model of a single cell crawling. And so we have to think about a framework that's generic enough that it can encapsulate all of this level of complexity. And I think that's quite difficult to do. When we're going to build models, we have to think about the levels of model abstraction. We need at least, to begin with, a conceptual model that's pretty close to biology, because you want biologists to be able to define things. You need to have some kind of mathematical or physical model that is a quantitative, unambiguous description of dynamics. And then, of course, you have to implement things as a simulation. I'm actually more and more convinced that the top two layers are the hard ones. If you could build a good conceptual model formalism, I think everything else becomes a little bit easier. But keeping that conceptual model, first, we don't have good ways of representing it. Second, keeping it connected to the more detailed specifications, I think, is tricky. Again, the networks modeling communities where you have graphical representations of networks coupled to a statement about interactions, and then coupled finally to instantiation as ODEs, for example, or Gillespie is a good analogy of what we need. We have a lot more model elements in terms of object categories. We have fields, we have cells, we have dynamic networks. There are other kinds of things that we also might have that we need. There are a whole variety of processes that have to be described. These obey different kinds of dynamics. There are also different events that occur in these situations. Cell differentiation, cell division, death, and disappearance. So we have to deal with reorganization of our agents, changes of shape. We have to deal with creation and destruction and change of type of agents. These are things that are typical of all agent-based modeling, but are not something that we think about so much in network models. So again, we come back to this idea we need modular model specification. Biology seems very modular. We have concepts like individuals, organs, tissues, cells, molecules. We have processes like division, growth, circulation, chemotaxis. We have structural definitions like epithelial and tubules. And we'd like to be able to build computational instantiations, models, or at least formalize the description of these things in order to be able to build our models in a reusable way. The problem is that biology is rather fuzzy. Concepts like a cell type wind up being fiat concepts. They're not really intrinsic to the biology. Even surface concepts like the boundary of a cell, the actual surface of a cell, aren't always very well defined. In early embryos, you have partial cell division, where you have the nucleus reproduce, but the cells actually don't separate into distinct cells. Very often, whether you have one cell or two cell is not so obvious in a biological context. And the way that you're going to define those things and break them up is going to depend on the level of detail in the question you're asking. And so there isn't a single right answer. On the other hand, your modeling specification has to make decisions about that. So the first thing, as I say, is that we have a problem, which is that the exposed variables and parameters that we're going to use will depend on how we decide to represent our objects. If we have agents versus a continuum, we're going to have very different ways of describing, say, a virus density. Whether a cell has an explicit shape or a surface to which you can attach things is going to just change in a fundamental way the way we think about describing that cell. On the other hand, if we want our models to be portable, we don't want to be dependent on the specific implementation details. And I think that that's actually means it's even more important that we describe underlying biological objects and processes, behaviors, and interactions in a method-independent way, and then do all of that additional specification with model-specific metadata. Let's just think about trying to build a library of cell types to do this population of that experimental images that I showed at the beginning. Well, I'm going to have some library of cell types. I'll have maybe a T-cell model. I'll have a endothelial cell model. And I'd like those models, those cell type models, to be reusable. In a real experiment, if I bring two cells together that are living, they'll do something because I inherit physics and chemistry. However, at the moment, in the modeling environments that we have, none of those things can be taken for granted. I can't assume that Physicell, Morpheus, CompuCell, and TissueForge will have the same idea of what cells can do, what their dynamics are, what events are available. And so in general, if I tried to bring two objects together, even two objects that were defined within the same modeling environment, for different purposes, they're going to have incompatible APIs. They're going to assume certain information is available to them that's not going to be made available, and the simulation is going to crash. And so I have to pre-specify all the behaviors and processes that my object can participate in. And I need to, that's quite verbose. And that's why most VT modeling frameworks don't do it now. We were just hearing a talk earlier by Paul Macklin, who has been developing Physicell, which is a center-based model, for a long time. And he, I think, has made some really interesting advances in terms of defining what properties and behaviors need to be implemented by any modeling framework, and some simple ideas about how to name them and how to define their interactions with the outside world that I think could be adopted quite widely. Something that we do need is that you need defaults. If you are expecting certain information that's not available, you have to know what to do. And so you want to make sure that all of those defaults are built into your system. Processes are hard. Processes shouldn't be stateful, which makes their specification rather complicated. Processes don't change things by themselves. Processes tell a dynamics or a set of events what to do. And in a sense, in network models where you typically have rate equations, the dynamics is d by dt, which is reasonably trivial, less trivial if you're doing something like Gillespie. And so you have some time stepper that has to deal with multiple simultaneous processes. It's not always possible to tell from looking at the internal state of an object what processes it's participating in, because it depends on its context. And that makes life a little bit harder. Again, when you're dealing with agent-based modeling, you have to have some concept of the universe as an object itself that has the dynamics that are going to be acting on the components inside of it and are watching for events. And so I think that that's a tricky thing to do. That makes object privileged model specification actually rather tricky. You need contingencies. You need rules to specify when an interaction is active. You need something to deal with incompatible or clashing interaction declarations if you're going to be able to pull from a library. That's also not so easy. You have to have a set of basic spatial relations, what the sphere of influence of a particular process is, where the consequences of an interaction activate. And it's not so easy to do this in a consistent way. Hearing Paul's talk just now, he has a very beautiful set of rules which say if under condition X, change property Y. But as we know, when we're looking at things like gene regulatory networks, if you have a lot of signals that come together, the way those signals integrate may be quite complicated. It's not just an and or an or or an addition. And so building rules that are flexible enough to have complex multi-component interactions, I think, is pretty hard. We do need standardized descriptions for contingencies and spatial relations. We don't really have that yet, although I think it's possible to do that. Adjacency, interaction range, volume exclusion. We need unified terminology for understanding these concepts. Encapsulation is necessary computationally. We need objects, and we need to have interfaces. We need information to flow in and out of the object through these well-defined interfaces. In biological reality, because the actual physical boundaries of objects are fuzzy and crossed, it's not always easy to make that correspond. If we're going to have a list of cell types in a simulation, then every object will be of one type at any given time. I suppose we could define a set of membership probabilities. Things get a little bit messy. Biological boundaries are something that, again, seem quite benign, but become pretty difficult to deal with. Cell membranes are pretty complicated. Most models don't deal with the fact that cells actually have membranes and membranes are objects. Mathematically, we can't have two closed surfaces touch each other. On the other hand, most simulation methodologies treat boundaries as shared between two cells. So there are actually some built-in mathematical problems with that. On the other hand, I don't think, and I'm running out of time, so I'm going through a few of these ideas quite quickly. I do think that there are things we can do. We can start with a set of object classes that we agree on biologically. We can define a set of conceptual model specifications. This is essentially trying to define what I would call model structure, even if we can't agree on parameter translation. We can define units. That should be easy, at least. And I think Dan Cook's in the room, so he'll tell us how to say minutes, meters, seconds, and so on from OPB. We can agree that information only crosses object boundary through defined interfaces. That's already a step in the right direction. And then I think something that's really critical that maybe is a little bit more biological is that I think we can probably agree on a basic set of terminology and implement major cell processes and define fundamental concepts like rates. So we could define the phases of the cell cycle that we're going to say these are expected to be implemented in a model of this kind. We're going to have certain kinds of cell death, like apoptosis. We're going to expect those to be implemented. And if we start out by focusing on the rate at which things occur, that probably makes things a little bit easier because that gives us a well-defined interface. Forces and integration of forces and shape, I think, are harder to deal with. And those are going to vary more depending on implementation method. TJ has already talked, and I really deleted all the slide on software components. But something that I think we could think about would be more contract-based approaches, which is, I have to say, a term that I didn't know until I was at a conference in Colorado a few weeks ago, where all of our software modules include a description of what their capabilities are and how they interconnect. And so that the API description and the capabilities of the software, what it can do, are actually defined inside of the software and are introspectable. Ideally, we'd like to be able to do that also for our model components, but I think that that's a pretty heavy thing to be asking for. So I think to begin with, it would be nice to start by defining some simple use cases, focusing on these issues of really structure for modeling language. What data do we need to interchange between our model components? And I think that we haven't really talked about what cells do. We didn't have time in this today. Maybe we could come back and have that discussion another day. But I do think that it's time to sit down and do it. And I actually, again, talking to Paul Macklin, I'm really impressed that he has gone ahead and done it. It takes a lot of bravery to go ahead and define a set of rules and standards on your own in that way. And I think that working with those is probably a great way forward. Our group with Herbert and Paul have a little NSF grant now, which is to try to develop a open source community around these questions that we've been talking about today. And so we'd love to talk about trying to solve these problems together. CompuCell and TissueForge, of course, are both open source projects. They're not very well funded, but if somebody would like to volunteer to help us develop these kinds of standard ideas in that context, we'd love to talk to you. As I mentioned, I put it in the chat. We do have a virtual seminar series which talks about multiscale modeling. It has a rather strange name, which is viral pandemics and modeling because of its history. But I think looking at the people in the room, many people in this room might be interested in some of the talks that we have in that series, not so much on standards, but actually on applications, and might be willing to speak to us as well. And I'd like to thank our funders, and I hope that breakneck trot through a few ideas in this context will give us something to talk about. Thanks very much.
Thank you, James. Thank you, TJ. We have some time for questions and discussion, so please speak up.
I guess so I had my hand up. It's okay. You'll have to forgive me for being a newcomer to your field. I come from a pathology background with I'll call it a nerdism in imaging samples in a more complete way to get at the idea of three-dimensional modeling of cells in the context of the whole organism. In pathology, we deal with real life, and we have to see every cell type that's there. Modeling individual cell types, I find, as a pathologist, kind of useless. I need to see what this cell is doing to that cell, for example, in cancer, or this structure or basement membrane or whatever. So I've been frustrated trying to get to whole organism images where you have everything. So now we actually have the technology. I posted some links, and I just submitted today a grant to NSF about taking that imaging capability and then posting it somewhere, just like we used to when we started bioinformatics. We posted DNA sequence. Now we want to post images of organisms and develop tools for computational phenomics. Before it was tools for bioinformatics. We have to go, unfortunately, it's a lot more complex to go, as you guys well know, to image in 3D and 4D. So I need to talk to you guys because I am new to this, and I think we can only succeed if we can work together in some manner. So today's the first start for that. So forgive me for not knowing what you guys do that well, but I've been very frustrated with pathology and having only 2D. You look at 2D images to 5 microns thick, and that's pretty poor way to get 3D modeling of cells and tissue.
Well, there are two issues, one of which is that you're seeing a 2D section of a 3D object, which is very limiting because you can easily misunderstand the structures in 3D. Classic example of that was mitochondria, where an old textbook, the mitochondria, a little ball, and they discovered that they're actually mostly connected to each other because when you take the 2D section, you can't tell the difference.
Nope.
I did a lot of imaging in my lab. I shut my wet lab down at the beginning of COVID, but I did experiments for many years, so I appreciate that. The other thing, of course, is the moment you're talking about pathology, you're talking about dead samples, and there's some fundamental mathematics that says you cannot actually infer the time course of an individual from even an infinite number of separate fixed time samples. Sometimes you're lucky and you can do it, but sometimes you can't. And as I say, you can't necessarily observe what processes the cell is participating in from a static image. So there are a whole series of very fundamental questions that you've raised, some of which I have some ideas about, some of which I don't. But that's why I think it's so valuable to be able to have the idea of, here's an image of cells, I now have a library where it says this is a model of a T cell, this is a model of a B cell. I now put it in the spatial context. I program it with the omics data that you can now get from single-cell RNA-seq. And now I want to see run it. It's still not going to replicate exactly what would have happened in real world. But nevertheless, it's a start to try to do this. At least getting the spatial configurations is essential. Implementing the spatial configurations is actually not that bad. Importing these into a simulation environment like CompuCell or PhysiCell isn't too bad, or a tissue forge. But describing what those cell states are and what they're doing is hard. And I think we should talk, and I would love to talk about this more, because I think critically there are two directions, one of which is taking these simulations and mapping it to say, what would it look like if a pathologist did this? And then going the other direction, which is given what you can do with the images, what can you infer that you could use to constrain a model? I should say there's a whole other use of this kind of modeling, which is for transfer learning, where you build a simulation, say, of a tumor in a programming language, in a modeling environment like CompuCell. You then simulate what it would look like in a pathology section, and you use that then to train, say, an image segmentation algorithm or a machine learning pathologist type of pathology and analysis package, and you pre-train it with what the simulated experiment data sets. Because one problem in biology is there is no gold standard for machine learning, because everything is somebody's opinion. However, if you'd have a simulation, you actually do have a gold standard. It may not be the gold standard for the real biology, but it winds up that transfer learning that way is pretty powerful. I'm not a huge machine learning buff, but that's a place where the mechanistic modeling and machine learning can work together really well.
If I can cut in there, actually, one of the primary pieces of novelty that we are anticipating to get from this is that there needs to be biology informed question asking in machine learning. All the neural networks for 1 million years is never going to give you the insight that a pathologist has. I'm sorry, because we simply know a hell of a lot more than the neural networks do. And I want to, support your statement about the mitochondria and extend that to the space of, well, a lot of us are going to be susceptible to this. Prostate cancer is based on similarities to Gleason scores, which are from these 2D ink drawings of patterns of glands that are clearly 3D, but if they pretend that they're 2D, which is nonsense. So we actually want to generate 3D models, and good luck modeling a 3D gland, because we don't even know what the hell they look like yet. So I'm frightened to death of this whole thing, but I love being frightened a little bit. It's like skiing downhill. I don't know when I'm going to crash, but I think this is really a fun problem to try to attack and get some sense out of it. And I have to say that even though we're imaging dead tissue all day in my pathology department, we make diagnoses every day that actually are acted upon quite significantly for patients. And you can't predict, absolutistly, we can't predict things, but we do our best. And what I'd like to do is improve that and make things better for patients. We have a half a micron.
Keith, we have another question I want to make sure to get to.
So I'll just finish the sentence and say we have half micron steps, whereas histology is 5 micron steps. Leave it at that. Sorry about talking too long.
Jane, do you have a question?
Yes, thank you so much. I have a questions about from the previous two speakers, the concentration of chemical species, if you will, in the cell. How do we get the information about the concentrations and reaction rate, physiological reaction rate inside the cell, or when you have a disease, what happened to the reaction rate? How do we define them? Where do you get the data from? I'm coming from I work for Food and Drug Administration, and we receive QSP submission, quantitative system pharmacology submission, mechanistic modeling. It's really a lot of work to review the parameter, the literatures they cited, and to be able to understand whether the model, to assess whether the model is credible. And I will be very grateful if you can kind of share with me how you get those information, and that could help us assessing the mechanistic model that have been submitted to us. Thank you.
EJ, did you want to say something to that? I mean, this is a bit far from the theme of the discussion today. It's very important, like the pathology comments, they're very important and critical. And I think they illustrate how hard these problems are and also how central they are. Yesterday in that seminar series that I mentioned, we had a talk by someone from Certara. And one of the people asked the question, they're building a model of inflammatory bowel disease. Non-spatial model, pure ODE model of inflammatory bowel disease. And somebody asked the question, and it's not that complicated. It has maybe 30 variables, 40 variables in it. And somebody asked the question, how long did it take you to build the model? And the answer was they had 5 senior modelers for 3 years full-time. They had 20 senior experimentalists for 3 years full-time, plus their assistants, another 60 people, doing in vitro and organoid culture experiment to measure the parameters for that one model. So, and they, as they said in the talk yesterday, there's still a bunch of stuff they weren't able to identify. Now, that model is already predictive and useful for drug discovery and potentially for patient personalization. But I did a back of the envelope calculation. If you just look at that, the effort they put in, that's about $150 million to $200 million to that one model. Clearly, a small individual investigator group like mine or Herbert's aren't going to be able to do that kind of scale of work. And so we have to focus on the things I think that initially that we can do. And I'm not trying to duck your question, but I think that the question at the moment is pretty hard for us. One of the things that we get biologically, which is nice, is that biology tends to be pretty robust. And so one of the things that I used to say to students is that if changing the value of any parameter in your model by a factor of 4 makes a difference, the model is structurally wrong. Because the level of fluctuation that's natural in biological systems is so large that very often you don't have to get the numbers exactly right. You typically know whether the lifetime of a protein is a millisecond, a second, a minute, an hour, or a day. And so if you get those orders of magnitude right, then the parameter space in which you have to search to do your parameter identification is much less. And so I think it's really critical to have good, I talked about good defaults in the context of connecting things. Good defaults for parameterization are really important. Something like a diffusion constant. Well, we measure you measure the diffusion constant of a chemical in water, in cytoplasm, in gel. They're all going to be very different. And so then you move back to things that maybe you have more control over, more measurable, like diffusion length rather than diffusion constant. And so I think that there really is a problem, and actually the Certara people talked about this quite a bit, connecting the clinical observables. In fact, you were the previous question was talking about how do you connect these clinical observables seem to, for scoring prostate cancer, for example, don't connect very well to the molecular concentrations which you can measure. The molecular concentrations, because they're at fixed time points, don't give you the kinetics. You can measure equilibrium constants, but you can't measure forward and backward rates. And so I think that you're right. I don't have an answer for this except to say that I think that the more we can make the software work smoothly and interoperably, the easier it's going to be to build the structures that will allow this. In other words, at the moment, when the models of the kind that we're talking about are built, they're not reusable, they're thrown out. And so all of that complex work that's being done to build those models is lost every time somebody wants to reuse things. And that's really a pity, because it means that we can't gradually say, here is a parameter, we can only constrain it within a factor of 10, but we can work with that, and now we can do additional simulations which are able to constrain it more. And so I think actually interconnectability and reusability of modeling components is going to be a critical part of enabling what you're talking about simplification of parameter identification and validation. It's not an answer, but it's necessary.
And thank you so much to James and TJ. We're over time right now, but perhaps if you're willing to wait for one more question before we go. Does anyone have one last question? It seems to me, I mean, you're talking about the scale that's required for individual modeling efforts becomes such a burden on the field, that if we are able to support smaller efforts and combine them and reuse them, then that's certainly a step in the right direction. But again, fascinating talks. Stay tuned for the next in the seminar series. We don't have them scheduled yet, but you'll be getting an email. Again, thanks to our speakers, and have a good day, everyone.